Welcome to The Sleepy Pharmacist. This channel shares calm, factual information about medicines and pharmacology. In this video, you'll hear quiet facts about drug interaction mechanisms drawn from real medications, enzymes, and historical cases. You can listen actively or let the information fade into the background as you rest. The Cytochrome P450 enzyme superfamily is the dominant phase 1 metabolic system for xenobiotics in humans. Phase 1 metabolism includes oxidative, reductive, and hydrolytic biotransformation reactions. The Cytochrome P450 enzyme superfamily is the dominant phase 1 metabolic system for xenobiotics in humans. Phase 1 metabolism includes oxidative, reductive, and hydrolytic biotransformation reactions. Cytochrome P450 enzymes are primarily responsible for oxidative drug metabolism. Cytochrome P3A4 metabolizes approximately 50% of drugs undergoing hepatic biotransformation. Cytochrome P3A4 is expressed in hepatocytes of the liver and in enterocytes of the small intestine. Expression in enterocytes allows cytochrome P3A4 to participate in intestinal drug metabolism. First-pass metabolism occurs at both hepatic and intestinal sites for cytochrome P3A4 substrates. Intestinal cytochrome P3A4 contributes to pre-systemic metabolism before drugs reach systemic circulation. Intestinal cytochrome P3A4 activity can be selectively inhibited without affecting hepatic cytochrome P3A4 activity. The grapefruit juice phenomenon demonstrates selective inhibition of intestinal cytochrome P3A4. Cytochrome P2D6 exhibits extreme genetic polymorphism across human populations. In some populations, 5 to 10% of individuals are classified as cytochrome P2D6 poor metabolizers. Poor metabolizers of cytochrome P2D6 lack functional enzyme activity. Cytochrome P2D6 is not inducible by xenobiotics. This lack of inducibility distinguishes cytochrome P2D6 from most other major cytochrome P450 enzymes. Cytochrome P2D6 substrates include many compounds affecting monoamine neurotransmitter systems. Cytochrome P2C19 shows significant genetic polymorphism. Loss-of-function cytochrome P2C19 alleles are common in East Asian populations. Cytochrome P2C9 metabolizes compounds with narrow therapeutic indices. Certain anticoagulant drugs are substrates of cytochrome P2C9. Cytochrome P1A2 is inducible by polycyclic aromatic hydrocarbons. Polycyclic aromatic hydrocarbons are present in cigarette smoke. Cytochrome P2E1 metabolizes small organic molecules. Ethanol is a substrate of cytochrome P2E1. Cytochrome P2E1 is inducible by chronic ethanol exposure. Enzyme induction occurs through transcriptional upregulation. This transcriptional upregulation is mediated by nuclear receptors. Nuclear receptors function as ligand-activated transcription factors. The pregnane X receptor, also called nuclear receptor 1I2, regulates drug metabolizing pathways. The pregnane X receptor is activated by structurally diverse xenobiotics. Activation of the pregnane X receptor drives transcription of cytochrome P3A4. The same activation also drives transcription of cytochrome P2C family enzymes. Pregnane X receptor activation increases transcription of various drug transporter genes. The constitutive androstenedione receptor, also called nuclear receptor 1I3, regulates detoxification pathways. The constitutive androstenedione receptor controls transcription of cytochrome P2B6. This receptor also participates in regulation of cytochrome P3A4 expression. The aryl hydrocarbon receptor responds to planar aromatic compounds. Activation of the aryl hydrocarbon receptor induces expression of cytochrome P1A family enzymes. Enzyme induction requires synthesis of new enzyme protein. This process involves transcription, messenger ribonucleic acid production, and protein translation. Enzyme induction develops gradually over days to weeks after exposure. Maximum induction typically requires 10 to 14 days of continuous exposure. Offset of enzyme induction begins after removal of the inducing compound. Loss of induction occurs over days to weeks rather than immediately. Existing enzyme protein must be cleared through normal protein turnover. Rifampicin is a potent activator of the human pregnane X receptor. Activation by rifampicin increases expression of cytochrome P3A4. Rifampicin-induced enzyme expression reduces systemic exposure of sensitive drug substrates. The magnitude of induction can substantially alter drug metabolism. These effects persist until newly synthesized enzymes are degraded. These pathways are dynamic rather than fixed. Their behavior depends on exposure, duration, and context. The next section shifts focus to how these changes develop over time and how they shape drug interactions in practice. As drug metabolism unfolds over time, additional mechanisms shape how enzyme activity increases, decreases, or is competitively altered in the presence of other compounds. Rifampicin induces expression of cytochrome P2C9. Rifampicin also induces expression of cytochrome P2C19. Rifampicin increases expression of the efflux transporter known as P-glycoprotein. Enzyme induction from rifampicin can reduce systemic exposure of sensitive drug substrates. In some settings, systemic exposure reductions reach 80 to 90%. St. John's wort is botanically designated Hypericum perforatum. St. John's wort contains the active compound hyperforin. Hyperforin activates the pregnane X receptor, a nuclear receptor regulating drug metabolism. Hyperforin-mediated enzyme induction occurs through mechanisms similar to those observed with rifampicin. The magnitude of induction from St. John's wort is generally lower than that seen with rifampicin. Autoinduction describes a process in which a drug increases metabolism of itself. Autoinduction leads to declining plasma concentrations over time. These declines can occur despite constant dosing. Carbamazepine is a well-known example of autoinduction. Carbamazepine induces cytochrome P3A4. Induction of cytochrome P3A4 increases carbamazepine clearance. Carbamazepine plasma concentrations typically decline during the first 2 to 4 weeks of therapy. Competitive enzyme inhibition occurs when an inhibitor occupies the enzyme active site. Competitive inhibitors compete directly with substrates for enzyme binding. The magnitude of competitive inhibition depends on inhibitor concentration. Binding affinity also influences the degree of competitive inhibition. Competitive inhibition begins immediately after inhibitor exposure. Competitive inhibition is reversible once the inhibitor is eliminated. Non-competitive inhibition involves inhibitor binding outside the enzyme active site. This form of inhibition reduces overall catalytic capacity. Reduced catalytic capacity occurs independently of substrate concentration. Mixed inhibition combines features of competitive and non-competitive mechanisms. Mixed inhibition patterns are observed with certain compounds. Mechanism-based inhibition is also called time-dependent inhibition. Mechanism-based inhibition is sometimes referred to as suicide inhibition. In this process, the enzyme converts the inhibitor into a reactive intermediate. The reactive intermediate irreversibly modifies the enzyme structure. Recovery from mechanism-based inhibition requires synthesis of new enzyme protein. Functional inhibition can persist longer than the inhibitor remains in plasma. Grapefruit juice contains compounds known as furanocoumarins. Bergamotin is one furanocoumarin present in grapefruit juice. 6'7' dihydroxybergamotin is another grapefruit-derived furanocoumarin. Grapefruit furanocoumarins act as mechanism-based inhibitors of intestinal cytochrome P3A4. Ketoconazole is a potent inhibitor of cytochrome P3A4. Itraconazole is also a potent inhibitor of cytochrome P3A4. Ketoconazole inhibits cytochrome P3A4 through direct enzyme binding. Itraconazole inhibits cytochrome P3A4 through a similar direct binding mechanism. The imidazole nitrogen of azole antifungal drugs coordinates with the enzymes heme iron. Coordination with the heme iron blocks access of drug substrates to the catalytic site. These structural features explain the strong inhibitory potency of azole antifungal compounds. P-glycoprotein is an efflux transporter involved in drug disposition. P-glycoprotein is encoded by the gene adenosine triphosphate binding cassette subfamily B member 1. P-glycoprotein is also known as multidrug resistance protein 1. P-glycoprotein functions as an adenosine triphosphate-dependent transporter. P-glycoprotein is expressed at the apical membrane of intestinal enterocytes. At sites of absorption, distribution, and elimination, transporter expression further shapes how drugs move through the body. P-glycoprotein is expressed at the canalicular membrane of hepatocytes. P-glycoprotein is also expressed at the luminal membrane of renal proximal tubule cells. Expression of P-glycoprotein is present at the luminal surface of brain capillary endothelium, forming part of the blood-brain barrier. P-glycoprotein limits oral drug absorption in the intestine. P-glycoprotein promotes biliary excretion of its substrates. P-glycoprotein also promotes renal excretion of substrates. Activity of P-glycoprotein restricts drug penetration into the central nervous system. Substrates of P-glycoprotein show wide structural diversity. Many P-glycoprotein substrates are lipophilic molecules. Many P-glycoprotein substrates also have amphipathic properties. Inhibition of intestinal P-glycoprotein increases oral drug bioavailability. Inhibition of P-glycoprotein at the blood-brain barrier increases penetration into the central nervous system. P-glycoprotein shares substantial substrate overlap with cytochrome P3A4. P-glycoprotein and cytochrome P3A4 are frequently co-regulated by the pregnane X receptor. Enzyme inducers often affect both P-glycoprotein and cytochrome P3A4. Enzyme inhibitors often affect both P-glycoprotein and cytochrome P3A4. Organic anion transporting polypeptide 1B1 is a hepatic uptake transporter. Organic anion transporting polypeptide 1B3 is also a hepatic uptake transporter. Organic anion transporting polypeptide 1B1 is expressed on the sinusoidal membrane of hepatocytes. Organic anion transporting polypeptide 1B3 is expressed on the sinusoidal membrane of hepatocytes. The sinusoidal membrane of hepatocytes is also referred to as the basolateral membrane. Organic anion transporting polypeptides facilitate hepatic uptake of anionic compounds. Uptake occurs from portal blood into hepatocytes. Inhibition of organic anion transporting polypeptides reduces hepatic drug uptake. Reduced hepatic uptake leads to decreased hepatic clearance. Decreased hepatic clearance results in increased systemic drug exposure. Some compounds inhibit organic anion transporting polypeptide 1B1 at portal vein concentrations. Inhibition at portal vein concentrations can produce clinically meaningful exposure changes. Organic anion transporter 1 is a renal tubular transporter. Organic anion transporter 3 is also a renal tubular transporter. Organic anion transporter 1 mediates renal secretion of anionic compounds. Organic anion transporter 3 also mediates renal secretion of anionic compounds. Organic cation transporter 2 is involved in renal secretion of cationic compounds. Organic cation transporter 2 mediates tubular secretion in the kidney. Inhibition of renal transporters reduces renal drug clearance. Reduced renal clearance prolongs the elimination half-life. Probenecid inhibits organic anion transporter-mediated renal secretion. Probenecid was historically used to prolong penicillin exposure. Breast cancer resistance protein is abbreviated as BCRP. Breast cancer resistance protein is encoded by the gene adenosine triphosphate binding cassette subfamily G member 2. Breast cancer resistance protein functions as an efflux transporter. Breast cancer resistance protein is expressed at the intestinal brush border. Breast cancer resistance protein is expressed at the hepatocyte canalicular membrane. Breast cancer resistance protein is also expressed at additional tissue sites. Breast cancer resistance protein shares overlapping substrate specificity with P-glycoprotein. Breast cancer resistance protein also has distinct substrate specificity. Intestinal barrier function involves coordinated activity of cytochrome P3A4 and P-glycoprotein. Cytochrome P3A4 and P-glycoprotein act together within enterocytes. A drug absorbed into an enterocyte may be metabolized by cytochrome P3A4. A drug absorbed into an enterocyte may be effluxed back into the gut lumen by P-glycoprotein. efflux back into the gut lumen allows repeated opportunities for cytochrome P3A4 metabolism. A drug absorbed into an enterocyte may also be transported into the portal circulation through the portal vein. The combined activity of cytochrome P3A4 and P-glycoprotein amplifies the intestinal barrier effect beyond either component alone. The grapefruit juice effect shows selective inhibition of intestinal cytochrome P3A4 without equivalent hepatic inhibition. Inhibition of intestinal cytochrome P3A4 increases oral bioavailability by reducing intestinal first-pass metabolism. The effects of grapefruit juice can persist for up to 24 hours after consumption. This persistence reflects mechanism-based inhibition that requires new enzyme synthesis for recovery. At the level of receptors, interactions are governed by shared binding sites and signaling mechanisms. Competitive antagonism at receptors follows mass action principles involving concentration and affinity. An agent present at higher concentration can displace another agent from a receptor. An agent with higher receptor affinity can also displace another agent from a receptor. Receptor occupancy determines the magnitude of pharmacodynamic effect. Two full agonists acting at the same receptor compete for binding. The net receptor effect depends on the relative intrinsic activity of competing agonists. The net receptor effect also depends on concentrations achieved at the receptor site. Partial agonists exhibit ceiling effects with submaximal receptor activation. Partial agonists can displace full agonists from receptor binding sites. In the presence of full agonists, partial agonists can function as antagonists. Buprenorphine demonstrates partial agonist properties at the opioid mu receptor. Some interactions occur through sites distinct from the primary binding domain. Allosteric modulation involves binding at a site distinct from the primary ligand binding site. Allosteric binding alters receptor conformation and modifies responses to orthosteric ligands. Positive allosteric modulators enhance receptor responses. Negative allosteric modulators reduce receptor responses. Benzodiazepines act as positive allosteric modulators at the gamma-aminobutyric acid type A receptor. Benzodiazepines enhance the effect of gamma-aminobutyric acid without directly activating the receptor. Over time, receptor systems adapt to sustained stimulation or blockade. Receptor upregulation represents a homeostatic adaptation to prolonged antagonist exposure. Receptor downregulation represents a homeostatic adaptation to prolonged agonist exposure. Chronic agonist exposure commonly produces receptor downregulation through internalization and reduced synthesis. Chronic antagonist exposure can produce receptor upregulation. Receptor regulation adaptations contribute to tolerance phenomena. Receptor regulation adaptations also contribute to withdrawal phenomena. Many clinically relevant interactions arise from convergence on shared neurotransmitter systems. Serotonergic mechanisms span multiple pharmacologically unrelated drug classes. Some antidepressants inhibit serotonin reuptake at the serotonin transporter. Monoamine oxidase inhibitors prevent enzymatic degradation of serotonin. Tramadol releases serotonin in addition to opioid receptor activity. Ondansetron and related compounds block serotonin 5-HT3 receptors as antiemetics. Triptans act as serotonin 5-HT1B and 5-HT1D receptor agonists. Linezolid is an oxazolidinone antibacterial with monoamine oxidase inhibitor activity as an off-target effect. Multiple serotonin-enhancing mechanisms can converge across drug classes, producing additive serotonergic activation. Similar overlap occurs within dopaminergic pathways. Dopaminergic overlap occurs across several therapeutic categories. Some antipsychotics block dopamine D2 receptors. Metoclopramide blocks dopamine D2 receptors and is also used as an antiemetic. Shared dopamine receptor targets explain movement-related effects across different indications. Cholinergic pathways also contribute to cumulative interaction effects. Anticholinergic activity involves blockade of muscarinic acetylcholine receptors. Anticholinergic activity is present in older histamine H1 antihistamines. Anticholinergic activity is present in tricyclic antidepressants. Anticholinergic activity occurs in certain antipsychotics. Anticholinergic activity is present in bladder antispasmodic agents. Anticholinergic activity is also present in some anti-Parkinsonian agents. Cumulative anticholinergic burden reflects additive effects at central and peripheral muscarinic receptors. Gamma-aminobutyric acid mechanisms converge at the GABA-A receptor complex, containing multiple distinct binding sites. Benzodiazepines modulate the function of the gamma-aminobutyric acid type A receptor complex in the central nervous system. Barbiturates also modulate gamma-aminobutyric acid type A receptor function through distinct binding sites. Ethanol modulates gamma-aminobutyric acid type A receptor activity as part of its central nervous system effects. Certain anesthetic agents modulate gamma-aminobutyric acid type A receptors through additional receptor domains. Different agents bind to distinct sites within the gamma-aminobutyric acid type A receptor complex. Simultaneous occupation of multiple sites can produce additive or synergistic central nervous system depression. Beyond receptor systems, interactions also emerge at the level of drug metabolism. Multiple cytochrome P3A4 substrates can compete for enzyme binding sites. Competition reduces metabolism of cytochrome P3A4 substrates. A compound with higher binding affinity preferentially occupies the cytochrome P3A4 enzyme. A compound present at higher concentration also preferentially occupies the enzyme. Preferential enzyme occupation reduces clearance of competing substrates. The magnitude of an interaction depends on the fraction of clearance mediated by cytochrome P3A4. Renal elimination pathways introduce additional sites for interaction. Drugs eliminated by the same renal transporter can compete for tubular secretion. Competition for renal tubular secretion alters pharmacokinetic behavior. Metformin is eliminated through organic cation transporter-mediated renal secretion, primarily involving organic cation transporter 2. Co-administration with organic cation transporter substrates or inhibitors alters metformin pharmacokinetics. Some interactions involve recycling processes between organs. Enterohepatic recirculation involves cycling of drug-related compounds between the liver and the intestine. Alteration of intestinal flora can interrupt enterohepatic recirculation. Binding of bile acids can also interrupt enterohepatic recirculation. Some antibiotics reduce bacterial beta-glucuronidase activity in the gut microbiome. Reduced beta-glucuronidase activity prevents deconjugation of glucuronide metabolites. Prevention of deconjugation interrupts enterohepatic recirculation. Several historically important cases illustrate how these mechanisms translate into clinical risk. The terfenadine-ketoconazole interaction was identified after regulatory approval and prompted increased focus on cytochrome-mediated interactions. Terfenadine is a prodrug requiring cytochrome P3A4-mediated metabolic conversion. The active metabolite of terfenadine is fexofenadine. Ketoconazole inhibits cytochrome P3A4 and prevents conversion of terfenadine to fexofenadine. Cytochrome P3A4 inhibition leads to accumulation of parent terfenadine. Parent terfenadine blocks the cardiac human ether a-GO-GO-related gene potassium channel, affecting the IKR current. Blockade of the human ether a-GO-GO-related gene channel produces QT interval prolongation. Cardiac arrhythmias led to investigation of the terfenadine interaction. Terfenadine was subsequently withdrawn from the market. Fexofenadine does not require metabolic activation. A similar pattern was observed with other agents. Cisapride is a gastrointestinal prokinetic agent that was withdrawn after recognition of drug interactions. Cisapride interacts with cytochrome P3A4 inhibitors. Cisapride blocks cardiac human ether a-GO-GO-related gene potassium channels. Cytochrome P3A4 inhibition increases cisapride exposure and produces QT interval prolongation. Other interactions arose through non-cytochrome pathways. The sorivudine-fluoropyrimidine interaction occurred in Japan in 1993. Sorivudine produces a metabolite that inhibits dihydropyrimidine dehydrogenase. Dihydropyrimidine dehydrogenase is the rate-limiting enzyme in fluoropyrimidine degradation. Inhibition of dihydropyrimidine dehydrogenase by the sorivudine metabolite is irreversible. Enzyme inhibition causes accumulation of fluorouracil to toxic concentrations. Transporter and enzyme interactions can also combine to amplify risk. The cerivastatin-gem-fibrozil interaction involves inhibition of organic anion transporting polypeptide 1B1. Gem-fibrozil glucuronide inhibits organic anion transporting polypeptide 1B1 mediated hepatic uptake. Inhibition of hepatic uptake increases cerivastatin systemic exposure. Gem-fibrozil inhibits cytochrome P2C8. Cytochrome P2C8 participates in cerivastatin metabolism. Combined uptake inhibition and metabolic inhibition substantially increase cerivastatin exposure. Increased cerivastatin exposure contributed to cases of rhabdomyolysis. Earlier theories of interaction mechanisms were later reevaluated. Plasma protein binding displacement was historically emphasized as a major drug interaction mechanism. The importance of plasma protein binding displacement has since been largely reconsidered. Some drugs are eliminated by high clearance mechanisms that are limited primarily by hepatic blood flow through the liver. Displacement from plasma protein binding increases the unbound fraction of a drug in systemic circulation. An increased unbound fraction produces a transient rise in unbound drug concentration. For high-clearance drugs, these transient increases in unbound concentration are rapidly cleared by hepatic blood flow. Some drugs exhibit flow-limited hepatic clearance that depends on liver blood flow rather than intrinsic metabolic capacity. For flow-limited drugs, an increased unbound fraction proportionally increases clearance. These proportional increases in clearance restore steady-state unbound concentrations to baseline despite protein binding displacement. Clinically meaningful plasma protein binding interactions are relatively uncommon. When such interactions do occur, they usually require mechanisms beyond simple displacement alone. Beyond clearance and binding, additional interaction pathways emerge during drug metabolism. Uridine diphosphate glucuronidyltransferase. Enzymes catalyze conjugation with glucuronic acid during phase 2 metabolism. Uridine diphosphate glucuronidyltransferase 1A1 glucuronidates bilirubin. This same enzyme also glucuronidates various drug substrates. Uridine diphosphate glucuronidyltransferase 2B7 conjugates morphine. This enzyme forms morphine 6 glucuronide. The same enzyme also forms morphine 3 glucuronide. Uridine diphosphate glucuronidyltransferase enzymes can be induced by certain compounds. Phenobarbital induces multiple uridine diphosphate glucuronidyltransferase isoforms. Uridine diphosphate glucuronidyltransferase enzymes can also be inhibited. Valproic acid inhibits several uridine diphosphate glucuronidyltransferase isoforms. Probenecid inhibits glucuronidation of certain substrates. Interactions can also occur before drugs ever reach systemic circulation. Absorption interactions can arise through physicochemical mechanisms within the gastrointestinal lumen. Chelation occurs when metal cations form insoluble complexes with drug molecules containing specific functional groups. Tetracycline antibiotics contain carbonyl and carboxyl groups that participate in chelation. Fluoroquinolone antibiotics also contain carbonyl and carboxyl groups involved in chelation. Both divalent and trivalent metal cations participate in chelation reactions. Calcium can form chelates with certain drug molecules. Magnesium can form chelates with certain drugs. Aluminum can form chelates with certain drugs. Iron can also form chelates with susceptible drug molecules. Chelation produces insoluble drug metal complexes with negligible oral absorption. Chelation occurs within the gastrointestinal lumen without involvement of receptors or enzymes. Gastrointestinal conditions further influence drug absorption and stability. Gastric pH influences drug dissolution. Gastric pH also affects drug stability. Some drugs require acidic conditions for adequate dissolution. Elevated gastric pH reduces dissolution of acid-dependent drugs. Reduced dissolution leads to lower oral absorption. Some prodrugs require acid-catalyzed activation. Elevated gastric pH reduces acid-catalyzed prodrug activation. Some compounds are unstable under low pH conditions. Acid suppression can improve stability of acid-unstable compounds. At the level of cardiac electrophysiology, additional interaction risks become apparent. QT interval prolongation can result from blockade of the human ether-agogo-related gene potassium channel. The human ether-a-go-go-related gene potassium channel mediates the IKR current in cardiac repolarization. Drugs from unrelated pharmacological classes can block human ether-a-go-go-related gene channels. The human ether-a-go-go-related gene channel contains a promiscuous binding cavity that accommodates diverse molecular structures. Certain antihistamines block human ether-a-go-go-related gene potassium channels. Certain antibiotics also block human ether-a-go-go-related gene channels. Macrolide antibiotics are known human ether-a-go-go-related gene channel blockers. Fluoroquinolone antibiotics can also block human ether-a-go-go-related gene channels. Certain antipsychotic agents block human ether-a-go-go-related gene channels. Certain antiarrhythmic agents block human ether-a-go-go-related gene potassium channels. Blockade of the human ether-a-go-go-related gene potassium channel produces prolongation of the QT interval on the electrocardiogram, also referred to as the ECG. QT prolongation effects from multiple human ether-a-go-go-related gene blocking agents are additive. Increased systemic drug exposure amplifies the effects of human ether-a-go-go-related gene channel blockade. Inhibition of cytochrome. P450 enzymes can increase systemic exposure of drugs that block the human ether-a-go-go-related gene channel. Inhibition of drug transporters can also increase systemic exposure of human ether-a-go-go-related gene-blocking drugs. Time-dependent factors influence the magnitude of drug interactions. The sequence of drug administration affects interaction magnitude. One drug can alter the disposition of another drug. Pre-administration of an inducer increases enzyme activity before exposure to a substrate. Maximal enzyme induction requires prior exposure to the inducing agent. Pre-administration of a mechanism-based inhibitor produces irreversible enzyme inactivation. This irreversible inactivation affects substrates administered afterward. Washout periods are required following enzyme induction. These washout periods account for normal enzyme protein turnover. Induced enzyme levels decline through standard protein degradation pathways. Washout periods are also required after mechanism-based inhibition. Recovery from mechanism-based inhibition requires synthesis of new enzyme protein. Pharmacovigilance systems rely on spontaneous adverse event reporting databases. The United States Food and Drug Administration maintains the Food and Drug Administration Adverse Event Reporting System, commonly known as FAERS. The European Union operates the EudraVigilance database. The World Health Organization maintains the VIGI-Base database. VIGI-Base contains over 40 million adverse event reports. VIGI-Base aggregates reports from more than 180 countries. Spontaneous reporting systems use standardized electronic data formats. The International Council for Harmonization E2B standard governs electronic case report transmission. E2B Revision 3 defines the current technical specifications. Pharmacovigilance databases use the medical dictionary for regulatory activities, commonly called MED-DRA, for adverse event classification. Spontaneous reporting systems are classified as passive surveillance systems. Passive surveillance systems lack denominator data. The absence of denominator data prevents direct calculation of incidence rates. Signal detection relies on disproportionality analysis rather than event frequency alone. The proportional reporting ratio is one commonly used disproportionality metric. A proportional reporting ratio threshold of 2 is often used for signal detection. The reporting odds ratio is another disproportionality metric. Reporting odds ratio signals require a lower 95% confidence interval above 1. The Bayesian confidence propagation neural network is a Bayesian signal detection method. This method generates information component scores. Information component scores identify drug event combinations that exceed expected frequencies. Multi-item gamma Poisson shrinker methods were developed by the United States Food and Drug Administration. These methods produce the empirical Bayes geometric mean metric. Gamma Poisson shrinker methods apply conservative shrinkage toward null values. This shrinkage reduces false positive signals when data are sparse. Signal detection methods differ in sensitivity. Reporting odds ratio sensitivity is approximately 0.55 in validation studies. Bayesian confidence propagation. Neural network sensitivity is approximately 0.53. Gamma Poisson shrinker sensitivity is approximately 0.23. Signal validation proceeds through defined sequential phases. Detection represents the first phase of signal validation. Validation follows detection as a distinct assessment step. Confirmation occurs after validation during signal evaluation. Analysis takes place after confirmation in the signal evaluation process. Assessment follows analysis as the next validation stage. Regulatory action occurs after the assessment phase. Each phase of signal validation applies separate evidentiary standards. Drug labeling changes in the United States are governed by statutory authority. The Food and Drug Administration Amendments Act of 2007 expanded post-approval regulatory authority for the United States Food and Drug Administration. Section 505(o) subsection 4 of the Federal Food, Drug, and Cosmetic Act authorizes mandated drug label changes. Changes being affected supplements allow immediate modification of drug labeling. These supplements do not require prior approval from the United States Food and Drug Administration. Post-approval safety labeling changes occur frequently. An analysis examined 278 new molecular entities. This analysis covered a period of 13 years. 70% of the analyzed new molecular entities received at least one new adverse reaction label addition. Drugs approved through expedited regulatory pathways show higher rates of post-approval labeling changes. Drugs approved through standard regulatory pathways show lower rates of post-approval labeling changes. Pre-approval clinical trials enroll relatively limited numbers of participants. Median pre-approval exposure is approximately 1,500 subjects. Cerivastatin was metabolized by both cytochrome P453A4 and cytochrome P452C8. The presence of dual metabolic pathways did not eliminate interaction risk. Gemfibrozil inhibited cytochrome P452C8 activity. The gemfibrozil glucuronide metabolite inhibited organic anion transporting polypeptide 1B1, a hepatic uptake transporter. This inhibition reduced hepatic uptake of cerivastatin. Reduced hepatic uptake increased systemic exposure to cerivastatin. Combined metabolic inhibition and transporter inhibition amplified exposure further. Elevated systemic exposure increased the risk of muscle toxicity. Post-withdrawal analysis showed that approximately 2% of users received contraindicated drug combinations. 1/3 of patients continued concurrent administration after withdrawal announcements. These findings demonstrated limitations of labeling-only risk mitigation strategies. The withdrawal period extended from approximately 1997 to 2001. Multiple drug withdrawals driven by interaction risk occurred during this era. Regulatory responses mandated earlier evaluation of drug interaction risk. In vitro reaction phenotyping became a required component of drug development. Clinical interaction studies with index perpetrator drugs were required. Screening for human ether-a-go-go-related gene potassium channel effects became standard practice. Interaction-driven drug withdrawals declined after these requirements were implemented. Pharmacovigilance systems depend on spontaneous adverse event reporting databases. The United States Food and Drug Administration maintains the Food and Drug Administration Adverse Event Reporting System, commonly known as FAERS. The European Union operates the EudraVigilance Reporting System. The World Health Organization maintains the VIGI-Base Global Safety Database. VIGI-Base contains more than 40 million adverse event reports. Reports in VIGI-Base originate from over 180 countries. Spontaneous reporting systems use standardized electronic reporting formats. The International Council for Harmonization, E2B standard governs electronic case report transmission. E2B Revision 3 defines the current technical requirements. Pharmacovigilance databases use the medical dictionary for regulatory activities, commonly called MedDRA, to classify adverse events. Spontaneous reporting systems are categorized as passive surveillance systems. Passive surveillance systems do not contain denominator data. The absence of denominator data prevents direct calculation of event incidence. Signal detection relies on disproportionality analysis rather than direct measurement of event frequency. The proportional reporting ratio represents one commonly used disproportionality metric. A proportional reporting ratio threshold of 2 is commonly used for signal detection within pharmacovigilance databases. The reporting odds ratio represents another disproportionality-based signal detection metric. Signals based on the reporting odds ratio require a lower 95% confidence interval greater than 1. The Bayesian confidence propagation neural network is a Bayesian method used for signal detection in large adverse event reporting systems. This network generates information component scores. Information component scores identify drug event combinations that occur more frequently than expected relative to background reporting. Multi-item gamma-Poisson shrinker methods were developed by the United States Food and Drug Administration. These methods generate the empirical Bayes geometric mean metric. Gamma-Poisson shrinker methods apply conservative shrinkage toward null values. This shrinkage reduces false positive signals when adverse event data are sparse. Signal detection methods vary in sensitivity. Reporting odds ratio sensitivity is approximately 0.55 in validation studies. Bayesian confidence propagation. Neural network sensitivity is approximately 0.53. Gamma-Poisson shrinker sensitivity is approximately 0.23. Signal validation proceeds through a series of defined phases. Detection represents the first phase of signal validation. Validation follows detection as a distinct assessment stage. Confirmation occurs after validation in the signal assessment process. Analysis takes place after confirmation. Assessment follows analysis as the next step. Regulatory action occurs after assessment is complete. Each signal validation phase applies separate evidentiary standards. Drug labeling changes in the United States are governed by statutory authority. The Food and Drug Administration Amendments Act of 2007 expanded post-approval regulatory authority for the United States Food and Drug Administration. Section 505 subsection 4 of the Federal Food, Drug, and Cosmetic Act authorizes mandated label changes. Changes being affected supplements allow immediate modification of drug labeling. These supplements do not require prior approval from the United States Food and Drug Administration. Post-approval safety labeling changes occur frequently. An analysis examined 278 new molecular entities. This analysis spanned a period of 13 years. 70% of the analyzed new molecular entities received at least one new adverse reaction label addition. Drugs approved through expedited regulatory pathways show higher rates of post-approval labeling changes. Drugs approved through standard regulatory pathways show lower rates of post-approval labeling changes. Pre-approval clinical trials enroll relatively limited numbers of participants. Median pre-approval exposure is approximately 1,500 subjects. Rare adverse events cannot be reliably detected at this exposure size. Events occurring at a frequency of 1 in 20,000 are unlikely to be observed before regulatory approval. The Weber effect describes characteristic adverse event reporting patterns after drug launch. Reporting rates typically peak 1 to 2 years after market introduction. Reporting rates decline following the initial peak period. The Weber effect biases detection toward acute adverse effects. Population expansion after approval introduces additional risk variables. Real-world use includes diverse metabolizer phenotypes. Real-world use involves polypharmacy combinations. Real-world use includes comorbidities excluded from clinical trials. Latency between exposure and adverse events complicates causal attribution. Passive surveillance performs poorly when temporal linkage between exposure and outcome is unclear. International Council for Harmonization Guideline E2A was established in 1994. ICH-E2A defined expedited adverse event reporting requirements. International Council for Harmonization Guideline E2B standardized electronic transmission of adverse event reports. International Council for Harmonization Guideline E2C evolved into what are now called Periodic Benefit Risk Evaluation Reports. Periodic benefit risk evaluation reports integrate both benefit assessment and risk assessment across a product's life cycle. International Council for Harmonization Guideline E2E introduced the concept of proactive pharmacovigilance planning. This pharmacovigilance planning framework applies before drug approval. Safety issues associated with terfenadine prompted major regulatory paradigm shifts. International Council for Harmonization Guideline S7b and International Council for Harmonization Guideline E14 were implemented in 2005. ICHS 7b mandates non-clinical evaluation of the QT interval. ICHE 14 mandates clinical thorough QT studies. The comprehensive in vitro proarrhythmia assay was developed as a successor framework. This assay evaluates multiple cardiac ion channels. The framework extends beyond assessment of the human ether agogo-related gene potassium channel alone. International Council for Harmonization Guideline M12 provides harmonized guidance for pharmacokinetic drug interaction studies. ICH-M12 was finalized in May 2024. Pharmacologic onset and offset are governed by different rate-limiting processes. Onset depends in part on the rate of distribution to the effect site. Distribution to the effect site is described by the effect site equilibration constant, Ke0. Onset also depends on receptor binding kinetics. Downstream signal transduction cascades further influence onset timing. Effect site equilibration half-life varies substantially between different drugs. Alfentanil has an effect site equilibration half-life of approximately 1.4 minutes. Fentanyl has an effect site equilibration half-life of approximately 3 to 4 minutes. Offset depends in part on the systemic elimination half-life of the drug. Dissociation from the effect compartment also contributes to offset. Turnover of physiological mediators further shapes offset timing. Distribution, elimination, and mediator turnover operate as independent processes. Counterclockwise hysteresis reflects delayed distribution to the effect site. This pattern can also reflect accumulation of active metabolites. Clockwise hysteresis reflects development of acute tolerance. Clockwise hysteresis may also reflect formation of antagonistic metabolites. Enzyme induction develops over characteristic and predictable time courses. Rifampicin produces detectable enzyme induction within approximately 24 hours. Maximal rifampicin induction typically requires 10 to 14 days. Phenobarbital has an elimination half-life of approximately 3 to 5 days. Phenobarbital induction effects generally appear after about 1 week. Enzyme induction requires activation of nuclear receptors. Activation of the pregnane X receptor initiates transcriptional signaling. Activation of the constitutive androstane receptor also initiates transcriptional signaling. Nuclear receptor activation leads to gene transcription. Gene transcription produces messenger ribonucleic acid. Messenger ribonucleic acid production leads to translation of enzyme protein. The rate of induction depends on inducer half-life relative to enzyme degradation half-life. Enzyme synthesis rate limits induction when the inducer half-life is short. Ligand persistence limits induction when the inducer half-life is long. Enzyme de-induction follows kinetics distinct from induction. De-induction depends on degradation of existing enzyme protein. Cytochrome P-453. A4 de-induction after rifampicin discontinuation has a half-life of approximately 7.7 days. Full recovery of cytochrome P-453. A4 activity may require 2 to 4 weeks. De-induction after discontinuation of St. John's wort occurs over approximately 46 hours. Cytochrome P-451A2 activity declines after smoking cessation. Cytochrome P-451A2 de-induction after smoking cessation has a half-life of approximately 39 hours. Cytochrome P-452E1 activity declines after alcohol withdrawal. The de-induction half-life of cytochrome P-452E1 is approximately 60 hours. De-induction of cytochrome P-452E1 is prolonged by ligand stabilization mechanisms. Intestinal P-glycoprotein returns to baseline more rapidly than hepatic enzymes. Recovery of intestinal P-glycoprotein occurs after approximately 14 days. Rapid enterocyte turnover accelerates intestinal enzyme recovery. Hepatic enzyme recovery is slower due to the longer lifespan of hepatocytes. Mechanism-based inhibition produces irreversible inactivation of enzyme molecules. This form of inhibition persists independently of drug plasma clearance. Recovery after mechanism-based inhibition requires de novo enzyme synthesis. 3 distinct modes of mechanism-based inhibition are recognized. One mode involves covalent binding to the cytochrome P450 enzyme apoprotein. Another mode involves alkylation or degradation of the cytochrome P450 hemiporphyrin. A third mode involves quasi-irreversible formation of a metabolite intermediate complex. Recovery time approximates 5 drug half-lives plus 5 enzyme half-lives. Repeated dosing amplifies the effects of mechanism-based inhibition. Each dose cumulatively inactivates additional enzyme molecules. Hepatic cytochrome P453. A4 has a half-life ranging from 36 to 140 hours. Intestinal cytochrome P453A4 has a half-life of approximately 23 hours. Grapefruit juice inhibits intestinal cytochrome P453A4 through a mechanism-based process. The effects of grapefruit juice can persist for 3 to 7 days after exposure. The sequence of drug administration can alter interaction magnitude. Sequence effects arise when one drug alters the disposition of another. Competitive protection occurs when reversible inhibitors block access of irreversible inactivators. This protection reduces the extent of effective enzyme inactivation. Absorption interference can occur through time-dependent chelation effects. Chelation prevents absorption when interacting drugs are present together in the gastrointestinal lumen. Mixed inhibitor-inducer compounds produce time-dependent shifts in interaction effects. Rifampin exhibits acute inhibition of certain drug transporters. Rifampin also produces chronic induction of metabolic enzymes. Rifampin may initially increase systemic exposure before reducing exposure over time. The dominant interaction mechanism can reverse with continued exposure. Enzyme saturation by one compound reduces metabolic capacity for subsequent compounds. Order of exposure determines the enzymatic capacity available for metabolism. Wash-in describes progression toward a steady-state effect. Steady-state concentration is reached after approximately 4 to 5 drug elimination half-lives. 4 to 5 half-lives correspond to roughly 94 to 97% of steady state. Wash-in of enzyme induction is delayed by the requirement for new enzyme synthesis. Maximum induction occurs after steady-state drug concentrations are achieved. Washout kinetics vary depending on the interaction mechanism. Reversible inhibition dissipates over approximately 5 inhibitor half-lives. Irreversible inhibition persists beyond clearance of the inhibiting drug. Recovery from irreversible inhibition requires both drug elimination and enzyme protein turnover. Enzyme induction shows slow wash-in and slow washout kinetics. Enzyme degradation governs the timing of induction washout. Intestinal enzyme recovery occurs faster than hepatic enzyme recovery. Differences in cell turnover rates explain tissue-specific recovery speeds. Cytochrome P453A4 and P-glycoprotein co-localize within enterocytes. This co-localization occurs at mature enterocytes located at villus tips. Enterocyte cytochrome P453A4 content exceeds that of paired hepatic specimens. Enterocyte cytochrome P453A4 levels are approximately 3 times higher than those observed in the liver. Enterocyte P-glycoprotein levels are approximately 7 times higher than hepatic levels. P-glycoprotein efflux recycles drug substrates back into the intestinal lumen. This recycling creates repeated opportunities for cytochrome P453A4-mediated metabolism. P-glycoprotein activity limits intracellular substrate concentrations within enterocytes. Limiting intracellular concentration helps prevent saturation of cytochrome P453A4. Approximately 50 to 60% of cytochrome P453A4 substrates are also substrates of P-glycoprotein. Dual inhibition of cytochrome P453A4 and P-glycoprotein produces multiplicative perturbation effects. Multiplicative effects exceed those expected from simple additive interactions. Oral bioavailability is determined in part by the fraction of drug that is absorbed. Oral bioavailability is also determined by the fraction that escapes gut wall extraction. An additional determinant is the fraction that escapes hepatic first-pass extraction. Metabolism within the gut wall can extract a substantial portion of an administered drug dose. Gut wall extraction may approach 40 to 50% for certain substrates. Midazolam demonstrates gut wall extraction comparable to hepatic extraction. Cytochrome P453, A4 expression is highest in the proximal intestine. The duodenum exhibits higher cytochrome P453, A4 expression than more distal intestinal regions. The jejunum also shows higher cytochrome P453A4 expression than the distal intestine. Beyond the intestine, transport processes shape tissue exposure. P-glycoprotein at the blood-brain barrier forms an additional extraction barrier. P-glycoprotein located on brain capillary endothelium limits penetration into the central nervous system. Knockout models lacking P-glycoprotein demonstrate marked increases in brain drug exposure. In these models, brain-to-plasma concentration ratios can increase by more than 2,000%. At the molecular level, structural features explain this breadth of interaction. Cytochrome P453, A4 possesses the largest active site volume among human cytochrome P450 enzymes. The apo active site volume of cytochrome P453 A4 is approximately 950 cubic angstroms. Ligand binding expands the cytochrome P453 A4 active site. The expanded active site volume may reach approximately 1,600 to 2,000 cubic angstroms. Cytochrome P453A4 accommodates substrates across a wide molecular weight range. Substrate molecular weights range from approximately 330 to 1,200 Daltons. The F-F'-G'-G helical region enables substantial conformational flexibility. A phenylalanine-rich cluster contributes to adaptable hydrophobic binding. Cytochrome P453 A4 can bind more than one ligand simultaneously. Simultaneous ligand binding produces heterotropic cooperativity. Heterotropic cooperativity results in non-Michaelis-Menten metabolic kinetics. Cytochrome P453 A4 accounts for approximately 30% of total hepatic cytochrome P450 content. Interindividual variability in cytochrome P453 A4 expression approaches 30-fold. Median cytochrome P453 A4 expression is higher in women than in men. Comparable structural flexibility is present in key transporters. P-glycoprotein contains a large internal substrate binding cavity. The internal cavity volume of P-glycoprotein is approximately 6,000 cubic angstroms. This cavity is lined with aromatic and hydrophobic amino acid residues. P-glycoprotein can bind multiple substrates at the same time. A 75 residue linker enables extensive conformational flexibility. Conformational switching allows inward-facing and outward-facing transporter states. P-glycoprotein portals open toward the cytoplasm. Additional portals open toward the inner leaflet of the cell membrane. This portal orientation enables direct extraction of substrates from cellular membranes. These coordinated features are regulated upstream. The pregnane X receptor, also known as nuclear receptor 1I2, regulates multiple detoxification pathways simultaneously. Activation of the pregnane X receptor induces phase 1 metabolic enzymes. Induced phase 1 enzymes include cytochrome P453A4. Induced phase 1 enzymes also include cytochrome P452C9. Cytochrome P452B6 is another phase 1 enzyme induced by activation of the pregnane X receptor. Activation of the pregnane X receptor, also known as nuclear receptor 1I2, also induces phase 2 conjugation enzymes. Phase 2 enzymes induced through this pathway include uridine diphosphate glucuronidyltransferase enzymes. Sulfotransferase enzymes are also among the phase 2 enzymes that are induced. Glutathione S-transferase enzymes are included within the induced enzyme set. Activation of the pregnane X receptor also induces multiple drug transporters. Transporters induced include multi-drug resistance protein 1, also known as adenosine triphosphate binding cassette subfamily B member 1. Multi-drug resistance associated protein 2, also known as adenosine triphosphate binding cassette subfamily C member 2, is also induced. Induction extends to members of the organic anion transporting polypeptide family. The constitutive androstenedione receptor, also called nuclear receptor 1I3, shares regulatory targets with the pregnane X receptor. The pregnane X receptor and the constitutive androstenedione receptor physically interact. Through this interaction, the receptors can mutually inhibit each other's activity. When unliganded, the pregnane X receptor acts as a net transcriptional repressor. Receptor crosstalk alters overall pathway output. Models lacking the pregnane X receptor demonstrate compensatory enzyme upregulation. Compensation by the constitutive androstenedione receptor occurs in the absence of pregnane X receptor activity. These regulatory principles became clinically visible through real-world outcomes. Terfenadine was identified as a prodrug that required metabolic activation. Cytochrome P453A4 converted terfenadine into fexofenadine. Ketoconazole inhibited cytochrome P453A4 mediated metabolism of terfenadine. This inhibition led to accumulation of the parent terfenadine compound. Parent terfenadine blocked cardiac potassium channels. This blockade involved the human ether-a-go-go-related gene potassium channel, also known as the HERG channel. Blockade of the human ether-a-go-go-related gene channel prolongs the QT interval. Cardiac arrhythmias were reported following terfenadine exposure. The interaction was identified during post-approval clinical use. Withdrawal of terfenadine occurred between 1997 and 1998. Fexofenadine replaced terfenadine as the marketed compound. Fexofenadine does not require metabolic activation. The terfenadine case established safety pharmacology as a distinct discipline. Mandatory evaluation of the QT interval followed this regulatory event. Subsequent withdrawals reinforced similar themes. Mibefradil was withdrawn from the market in 1998. Mibefradil functioned as a mechanism-based inhibitor of cytochrome P453A4. Mechanism-based inhibition produces irreversible enzyme inactivation. Recovery from this inactivation requires synthesis of new enzyme protein. Mibefradil exhibited a low inhibition constant. Mibefradil also demonstrated a high inactivation rate constant. Mechanism-based inhibition extended interaction effects beyond drug clearance. Interaction risk persisted after plasma drug concentrations declined. Original labeling contraindicated co-administration with multiple drugs. Despite these warnings, real-world co-prescription continued. This case demonstrated the limits of labeling-based risk mitigation. Mibefradil illustrated the phenomenon of time-delayed interaction emergence. Another pathway revealed the role of non-host metabolism. Sorivudine was approved in Japan in 1993. Sorivudine was withdrawn shortly after its market introduction. 18 deaths occurred within approximately 40 days of approval. Sorivudine itself did not directly inhibit the enzyme involved. Instead, intestinal microbiota metabolized sorivudine to bromovinyluracil. Bromovinyluracil irreversibly inhibited dihydropyrimidine dehydrogenase. Dihydropyrimidine dehydrogenase is the rate-limiting enzyme for fluoropyrimidine degradation. Enzyme inhibition occurred through covalent modification. This covalent modification targeted a cysteine residue on the enzyme. Inhibition produced a functional deficiency of dihydropyrimidine dehydrogenase, the primary enzyme responsible for fluoropyrimidine catabolism. This functional deficiency led to accumulation of fluoropyrimidine drugs. Toxicity resulted from elevated exposure to fluorouracil, a widely used fluoropyrimidine antimetabolite. The interaction occurred independently of hepatic metabolism. Gut microbiota acted as the mediator of this interaction. This event revealed drug interaction mechanisms driven by the human microbiome. Traditional pre-approval testing did not evaluate this metabolic pathway. The case expanded recognition of non-host contributors to drug metabolism. Regulatory frameworks later incorporated considerations related to the microbiome. A separate withdrawal highlighted transporter and enzyme overlap risks. Cerivastatin, a synthetic statin used for lipid lowering, was withdrawn from the market in 2001. The withdrawal followed reports of severe rhabdomyolysis. 52 deaths were associated with cerivastatin-related rhabdomyolysis. Cerivastatin demonstrated extreme pharmacologic potency compared with other statins. In vitro potency was approximately 100 times greater than that of lovastatin. Cerivastatin was metabolized by both cytochrome P453A4 and cytochrome P452C8. The presence of dual metabolic pathways did not prevent interaction risk. Gemfibrozil, a fibric acid derivative lipid-lowering agent, inhibited cytochrome P452C8. In 1954, a scientist named Martin Klingenberg observed an unusual absorption peak at exactly 450 nanometers. He made this observation while studying rat liver samples at the Johnson Research Foundation in Pennsylvania. The strange pigment Klingenberg identified did not match any known proteins described in scientific literature. Neither hemoproteins nor copper proteins produced a similar spectroscopic signature. When researchers attempted to isolate the mysterious pigment, it consistently disappeared. Any treatment used to dissolve it caused the distinctive absorption spectrum to vanish completely. In 1960, a scientist named Tsuneo Omura joined a laboratory at Osaka University in Japan. His work with rabbit liver samples led to a major breakthrough in understanding the pigment. Omura discovered that adding a substance called deoxycholate shifted the absorption peak from 450 to 420 nanometers. This shift resembled the behavior of a known cytochrome system. The original form was named P450, with the letter P standing for pigment. The converted form was called P420, reflecting its shifted absorption wavelength. A landmark paper published in 1964 established a method to measure the concentration of P450 enzymes. This publication has been cited more than 12,700 times. Between 1963 and 1965, researchers at the University of Pennsylvania demonstrated that P450 metabolizes drugs. Light-based experiments confirmed the enzyme's functional role. Before the late 1990s, drug interaction testing was largely optional during drug development. Regulatory agencies did not consistently require these studies before approval. A medication called Terfenadine was approved in 1985 as the first non-sedating antihistamine. It later became central to understanding dangerous drug interactions. By June of 1990, regulators identified serious heart rhythm problems in people taking terfenadine with certain antibiotics or antifungal medications. The pattern revealed an underlying metabolic mechanism. Terfenadine functions as a prodrug that is completely converted by the enzyme Cytochrome P3A4. The compound reaching the bloodstream is a different drug called fexofenadine. When Cytochrome P3A4 inhibitors blocked terfenadine's conversion, the parent compound accumulated in the body. This accumulation disrupted the heart's electrical system. Terfenadine received a black box warning in 1992. It was later withdrawn from the market between 1997 and 1998. After terfenadine was withdrawn, regulators approved its active metabolite fexofenadine as a separate medication. This preserved therapeutic benefit while removing the interaction risk. A calcium channel blocker called mibefradil was withdrawn in June 1998. It had been available for only 1 year before serious safety concerns emerged. Mibefradil caused dangerous interactions with at least 25 other medications. Its enzyme inhibition was unusually potent and long-lasting. Studies showed mibefradil destroyed approximately 70% of cytochrome P3A4 activity within the first minute of exposure. This rapid inactivation increased its hazard. An 83-year-old woman taking a cholesterol medication with mibefradil developed progressive muscle weakness. Her symptoms resolved only after 4 weeks without mibefradil. The prolonged recovery illustrated an important interaction principle. When enzymes are destroyed instead of blocked, the body must synthesize new proteins. A cholesterol-lowering drug called cerivastatin was withdrawn worldwide in 2001. Muscle breakdown events had been linked to its use. Of 31 deaths in the United States attributed to cerivastatin-related muscle damage, 12 involved concurrent gemfibrozil use. This accounted for 39% of fatal cases. Investigations showed gemfibrozil affected cerivastatin through 2 mechanisms. It blocked a liver uptake transporter and inhibited the enzyme responsible for breakdown. The cerivastatin case demonstrated that transport proteins require evaluation alongside metabolizing enzymes. Both systems can contribute to dangerous interactions. Health Canada received 31 reports of cerivastatin-associated muscle breakdown between March 1998 and June 2001. 9 cases involved gemfibrozil. The first formal drug interaction guidance from the United States Food and Drug Administration appeared in April 1997. It focused on laboratory testing methods. A complementary guidance addressing clinical studies followed in December 1999. Together, these documents formalized interaction assessment requirements. The 2006 draft guidance marked a major shift in the field. It included criteria for evaluating interactions involving transport proteins. That guidance introduced a mathematical ratio used to recommend clinical evaluation. This replaced earlier qualitative assessments. By 2012, guidance expanded to include multiple transporters and model-based evaluation approaches. Interaction assessment became more systematic. In January 2020, final guidance required evaluation of 9 key transport proteins. These included P-glycoprotein and several uptake transporters. In May 2024, the ICH-M12 guideline achieved international harmonization. Requirements were unified across the United States, Europe, and Japan. In 1976, Rudy Giuliano and Victor Ling identified a large protein in drug-resistant cells. The protein altered how drugs crossed cell membranes. The protein was named P-glycoprotein because it affected membrane permeability. The letter P stands for permeability, not phosphorylation. For nearly 2 decades, P-glycoprotein research focused on cancer. Scientists studied how tumor cells resisted chemotherapy drugs. A landmark study published in 1994 changed understanding of P-glycoprotein. Mice lacking the transporter showed increased drug sensitivity. These mice displayed 100-fold increased sensitivity to ivermectin. Drug concentrations in their brains were dramatically elevated. The findings showed P-glycoprotein was more than a cancer resistance mechanism. It acted as a fundamental gatekeeper for drug distribution. In intestinal cells, P-glycoprotein sits on the surface facing the gut interior. It actively pumps absorbed drugs back into the intestinal space. At the blood-brain barrier, P-glycoprotein limits drug entry into the central nervous system. Mice without it show brain levels 20 to 80 times higher. In kidney cells, P-glycoprotein promotes drug excretion into urine. In the placenta, it helps protect the developing fetus. An interaction between digoxin and quinidine was reported in 1968. The mechanism remained unclear for nearly 30 years. In 1996, research showed quinidine inhibited P-glycoprotein transport of digoxin. This explained 2 to 3-fold increases in blood levels. Digoxin is now the standard probe substrate for evaluating P-glycoprotein inhibition. Regulatory agencies rely on it during development. Cytochrome P3A4 and P-glycoprotein are both present in intestinal cells. Their proximity creates a coordinated absorption barrier. When a drug enters an intestinal cell, P-glycoprotein may expel it before metabolism occurs. This recycling extends exposure to cytochrome P3A4. The combined effect exceeds what either system produces alone. Pharmacologists describe this coordination as double jeopardy. Total intestinal cytochrome P3A protein measures about 70 nanomoles. Liver content reaches approximately 5,490 nanomoles. Despite this difference, intestinal extraction of the drug midazolam nearly matches hepatic extraction. High local concentrations explain the paradox. When drugs inhibit both cytochrome P3A4 and P-glycoprotein, exposure increases can become dramatic. Some combinations raise blood levels 7-fold or more. The antifungal medication ketoconazole inhibits both cytochrome P3A4 and P-glycoprotein at the same time. When combined with certain heart medications, blood levels have been observed to increase more than 7-fold. Cyclosporine inhibits multiple systems simultaneously, including cytochrome P3A4, P-glycoprotein, and several uptake transporters. Its effect on cholesterol medications can increase exposure 4 to 20 times depending on the drug. Grapefruit juice was discovered to cause drug interactions by accident. In 1989, researcher David Bailey used it to mask the taste of alcohol during a study. The active compounds in grapefruit juice are called furanocoumarins. These include bergamottin and a related compound known as 6'7' dihydroxybergamottin. Cytochrome P3A4 converts furanocoumarins into reactive molecules that permanently bind to the enzyme. This binding destroys the enzyme's ability to function. Normal grapefruit juice consumption affects only intestinal cytochrome P3A4. The furanocoumarins are removed from the bloodstream before reaching the liver in significant amounts. A single 200 milliliter glass of grapefruit juice produces effects lasting 3 to 7 days. Recovery requires replacement of intestinal cells and production of new enzyme. Mechanism-based inhibition differs fundamentally from reversible inhibition. When permanent bonds form with an enzyme, that enzyme molecule is destroyed. Recovery from mechanism-based inhibition depends on synthesis of new enzyme protein. This process is governed by the natural lifespan of enzymes within cells. Cytochrome P450 enzymes in the liver have a half-life of about 36 hours. Nearly 2 weeks may pass before normal function returns after stopping certain inhibitors. The antibiotic erythromycin undergoes a chemical reaction producing a compound that binds tightly to cytochrome P3A4. This creates a stable complex that inactivates the enzyme. A large study of Tennessee Medicaid recipients examined nearly 1.25 million person-years of data. Current erythromycin use was associated with double the risk of sudden cardiac death. When erythromycin was combined with cytochrome P3A4 inhibitors like diltiazem or verapamil, the risk increased 5-fold. The antibiotic also affects heart electrical channels directly. Regulatory guidance specifically addresses assessment of time-dependent inhibition during drug development. Special laboratory experiments are required to detect progressive enzyme loss. When time-dependent inhibition is detected, developers must determine 2 key values. These describe the maximum rate of enzyme destruction and the concentration producing half that rate. Guidance documents warn that separating drug doses does not prevent mechanism-based interactions. The example involves a stomach acid medication combined with a blood thinner. In 1993, an antiviral medication called sorivudine was approved in Japan. Within 40 days, 18 people died from severe bone marrow suppression. The deaths occurred in people taking sorivudine with cancer medications related to 5-fluorouracil. Gut bacteria converted sorivudine into a compound that destroyed a critical enzyme. The compound formed from sorivudine permanently modified an enzyme responsible for breaking down the cancer medication. This caused toxic accumulation of the drug. Sorivudine was never approved in the United States or Europe. The tragedy showed how gut bacteria can drive unexpected drug interactions. Unlike inhibition, enzyme induction requires days to weeks before observable effects emerge. The process relies on gene activation and protein synthesis. Inducers bind to specialized receptors inside cells and activate transcription of enzyme genes. The resulting messenger molecules are translated into functional protein. This multi-step process creates a predictable delay before induction becomes apparent. The lag phase can obscure recognition of interactions. The antibiotic rifampicin is considered a prototypical enzyme inducer. Maximum effect typically appears after 7 to 14 days of continuous use. St. John's wort requires 10 to 14 days to reach full inducing effect. Carbamazepine shows earlier effects but peaks after 1 to 2 weeks. A person starting rifampicin while taking oral contraceptives may not experience failure for 1 to 2 weeks. The delay can create a false sense of protection. When an inducer is stopped, accumulated enzyme must degrade naturally before function normalizes. This follows the enzyme's inherent lifespan. Hepatic cytochrome P3A4 has a half-life estimated between 70 and 140 hours. About 2 weeks may pass before baseline function returns. Intestinal cytochrome P3A4 has a shorter half-life of 12 to 33 hours. This reflects rapid turnover of intestinal lining cells. Differences between hepatic and intestinal enzyme half-lives carry clinical relevance. Recovery from induction occurs faster in the gut than in the liver. In early 2000, a study at the National Institutes of Health Clinical Center examined St. John's wort. It reduced blood levels of indinavir by 57%. The same study found 8-hour trough concentrations of indinavir dropped by 81%. These reductions permitted viral replication and resistance. The active compound in St. John's wort is hyperforin. It binds strongly to a cellular receptor known as the pregnane X receptor. Hyperforin induces cytochrome P3A4, cytochrome P2C9, cytochrome P2C19, and P-glycoprotein. This broad activity increases interaction potential. Following the indinavir findings, regulatory agencies issued coordinated warnings. Authorities in the United States, Canada, and the United Kingdom advised against combination use. Case reports documented transplant rejection in people taking St. John's wort with cyclosporin. The supplement increased metabolism below therapeutic levels. Other reports described breakthrough bleeding in women using oral contraceptives with St. John's wort. Hormone metabolism exceeded contraceptive thresholds. About 44% of adults aged 65 and older in the United States take 5 or more prescription medications. This prevalence has nearly doubled since the century's start. Among hospitalized older adults, 5 or more medications are used in 71% of cases. This level of polypharmacy increases interaction opportunities. Age-related physiological changes further raise interaction risk. Hepatic blood flow decreases by about 40% and liver mass by roughly 30%. Phase 1 metabolic capacity also declines with age. Older adults may experience stronger interaction effects than younger individuals. Prescribing cascades occur when a drug side effect is treated as a new condition. Additional medications may introduce further interaction risk. When calcium channel blockers cause ankle swelling, nearly 10% of patients receive diuretics. The swelling is a known effect of the original medication. When cholinesterase inhibitors cause urinary incontinence, bladder medications may be added. These drugs can worsen the cognitive symptoms being treated. For every 115 people starting certain antidepressants, 1 additional sleep medication is prescribed. This cascade effect appears in population studies. Cytochrome P2D6 shows at least 50-fold activity variation among individuals. More than 100 genetic variants have been identified. People lacking functional cytochrome P2D6 are called poor metabolizers. They represent about 7% of Europeans and 1% of Asians. Individuals with extra copies of the cytochrome P2D6 gene are ultra-rapid metabolizers. Rates reach 20 to 29% in East African populations. Ethiopia banned codeine due to population genetics. About 30% of Ethiopians rapidly convert codeine to morphine at dangerous rates. Inflammatory molecules in blood suppress cytochrome P450 activity in liver cells. Interleukin-1 beta, interleukin-6, and tumor necrosis factor alpha all contribute. A marker of inflammation called C-reactive protein shows an inverse relationship with drug clearance. Higher levels are associated with reduced activity of cytochrome P3A4, cytochrome P1A2, and cytochrome P2C19. In heart failure, hepatic blood flow can decrease by about 40%. Organ congestion further impairs the liver's ability to process medications efficiently. One study found propranolol clearance was reduced by 97% in people with right ventricular failure. This reduction far exceeded predictions from healthy volunteer studies. Drug interaction predictions based on healthy volunteers may underestimate effects in real patients. Disease states can alter metabolism in ways trials do not capture. Pre-approval clinical trials have inherent limits for detecting drug interactions. Trial populations are usually healthier than those who later receive the medications. Specific combinations of inhibitors and inducers are rarely tested before approval. The number of possible combinations exceeds what development programs can evaluate. Clinical trial sample sizes are often too small to detect rare interaction events. Some dangerous combinations affect only 1 in several thousand people. Fixed study durations may miss interactions emerging only with chronic therapy. Certain effects appear after months or years of combined use. A review of 278 new drug approvals found frequent post-marketing discoveries. 70% had at least 1 new adverse reaction added after release. The United States Food and Drug Administration Adverse Event Reporting System began in 1997. It was renamed in 2012 and continues collecting safety reports. Pharmaceutical manufacturers must submit adverse event reports to the database. Healthcare professionals and consumers may also report events voluntarily. Statistical algorithms analyze reports for potential safety signals. These include proportional reporting ratios and empirical Bayesian calculations. The World Health Organization maintains a global database called Vigibase at the Uppsala Monitoring Center. It contains over 40 million individual safety reports worldwide. The Uppsala Monitoring Center receives reports from 160 member countries. These nations represent nearly 99% of the global population. Pharmacovigilance systems detected the link between statins, gemfibrozil, and muscle breakdown. This signal contributed to cerivastatin's market withdrawal. During the early months of the COVID-19 pandemic, a new interaction signal appeared. Reports linked lopinavir, ritonavir, and atorvastatin to muscle breakdown. Spontaneous reporting systems have known limitations such as underreporting. Many adverse reactions are never submitted to regulatory databases. Selection bias affects which events are reported. Dramatic or unexpected reactions are more likely documented than subtle effects. Establishing true causation from spontaneous reports is challenging. Database association does not automatically prove causality. About 36% of indication data in the reporting system is missing. Incomplete records limit analytical conclusions. Despite limitations, pharmacovigilance databases remain essential for safety monitoring. They detect interactions impossible to predict before approval. Post-marketing data confirmed predictions involving statins and cytochrome P3A4 inhibitors. Adverse event rates were 6 times higher with combined use. The reporting rate for simvastatin with a cytochrome P3A4 inhibitor was 38.4 per 10 million prescriptions. Without inhibitors, the rate was 6 per 10 million. Current regulations require evaluation of whether new drugs are enzyme substrates. Developers must determine how compounds are metabolized. Developers must also assess whether new drugs inhibit key enzymes. A medication may be safe alone, but hazardous in combinations. The same evaluation applies to transport proteins. Drugs are assessed as both substrates and inhibitors of transporters. 9 transporters are named in current regulatory guidance. These include P-glycoprotein, breast cancer resistance protein, and organic transporters. Organic anion transporting polypeptide 1B1 is vital for liver drug uptake. Many cholesterol-lowering drugs depend on this transporter. Breast cancer resistance protein was first identified in chemotherapy-resistant cancer cells. It later proved important for normal drug distribution. Multi-drug resistance-associated protein 2 is found in liver cells facing bile ducts. It pumps drugs and metabolites into bile. Organic cation transporters handle positively charged drugs in the kidney. They contribute to elimination of medications like metformin. Multi-drug and toxin extrusion transporters work alongside organic cation transporters. Together, they support renal drug elimination. The ICHM-12 guideline established the first harmonized interaction assessment standard. Agencies in the United States, Europe, and Japan follow it. Before harmonization, requirements differed across regions. A study accepted by one agency might not satisfy another. Harmonized guidelines reduce duplicate studies across submissions. This efficiency can speed development without reducing safety oversight. Health Canada generally aligns interaction requirements with the United States Food and Drug Administration. It also performs independent safety reviews. Canadian regulators removed terfenadine from the market in 1999. This occurred 1 to 2 years after United States withdrawal. Canada participated in harmonization of the ICHM-12 guideline. National requirements now match the global interaction standard. The molar extinction coefficient established in 1964 remains in use. Researchers still apply 91 per millimolar per centimeter to quantify cytochrome P450. Spectroscopic methods from the 1960s shaped modern enzyme characterization. Techniques have evolved, but principles remain consistent. Early drug metabolism research relied on animal models like rats and rabbits. Human-based systems emerged gradually over time. Human liver microsomes became available as transplant programs expanded. Excess surgical tissue supported laboratory research. Recombinant enzyme systems allow isolated study of individual human cytochrome P enzymes. Single enzymes are expressed in cell lines. Human hepatocyte cultures preserve full liver metabolic machinery. They provide more realistic metabolism assessment than isolated enzymes. Moving from animal models to human systems improved prediction accuracy. Species differences had caused misleading early results. Rats express different proportions of cytochrome P enzymes than humans. Metabolic pathways can differ substantially between species. Cytochrome P2D6 is especially problematic for cross-species prediction. Rats lack a true equivalent of this human enzyme. Modern drug development integrates multiple predictive approaches. Laboratory studies, modeling, and clinical trials each contribute data. Physiologically based pharmacokinetic models simulate drug movement mathematically. These models incorporate enzyme and transporter properties. Regulatory agencies increasingly accept model-based predictions. In some cases, modeling reduces the need for clinical interaction studies.